Megan, and I'm a first-year PhD student in Dr. Garcia's lab in the bio department. I'm going to continue our neuroscience spiel for today by talking a little bit about immunohistochemistry. Has anyone used immunohistochemistry, or does anyone know what it is? A few nods, a few shakes. All right, so we'll go through it. Basically, it's a method of using antibodies, which we all have in our blood, right, to label peptides or specific neurons or any types of cells, actually. You can use it outside of the brain as well. And basically, we'll have a cell. Here's our cell, and cells produce lots of things that they release or have on the inside. There's a nucleus, right? It produces lots of things. And if we want to label this cell, and we know that it has a specific peptide, we can use antibodies that are purified from the blood of typically rodents or smaller animals, and label them onto here. So through immunohistochemistry, we are able to make the membrane permeable to our antibodies. So we have a primary antibody, and we have usually a secondary antibody. Does anybody who has used immunohistochemistry want to share what the difference is between these two? Sure.
The primary will bind directly to the antigen of interest, whereas the secondary will bind to the primary. And it's usually with a target.
That's exactly right. So our primary antibody, as she said, in case you guys didn't hear, will bind to our antigen of interest, or peptide, or whatever we want to label. And the secondary antibody will bind onto our primary antibody. So usually we'll have an antigen here, and we'll have our primary antibody here. We will attach. This is usually in the cell. And then the secondary antibody will attach onto that. So our secondary antibodies for neuroscience usually have a fluorescent tag on them to make them able to visualize under light microscopy, or fluorescent microscopy. We can also do a series of reactions with the secondary antibodies in order to make them a permanent stain. That's a little bit more complex. But basically, the method of using antibodies in neuroscience is useful because we can label specific cells of a specific phenotype, or we can inject a tracer into the brain, for example, and detect our tracer that has now been taken up by neurons of interest. We can also use multiple rounds of immunohistochemistry or different colored fluorescent tags to produce new phenotypes of the, or not new phenotypes, I'm sorry, co-localization of a peptide and a tracer in a neuron. So then that cell would be expressing more than one of these antigens of interest. Are there any questions? Yes?
So you said you can use these probes to bind to many different types of cells, right? So you could bind to like a certain phenotype or a certain peptide. So how do you design a probe to fit to a specific antigen?
Okay. Yeah, good question. So the primary antibody would be raised in, for example, a rat. We can use that primary antibody in a mouse model of neurons. So it'll be specifically made in the rat to bind to mouse tyrosine hydroxylase, for example. So this is going to be rabbit, anti-TH, right? It'll be tyrosine hydroxylase, so it'll bind that specifically to that. And then our secondary antibody would be raised in a different animal, so a sheep, and it would be anti-rabbit. Oh, I said rabbit to begin with, but we're going to do rabbit, sorry. So this sheep anti-rabbit will detect only the rabbit antigens in the entire brain. So if we're in a brain of mouse, it won't detect the rest of the cells. It'll specifically bind to the ones that have the rabbit. Did that answer your question?
Yeah, more or less.
Yes.
So how does that work? Like, how does the secondary recognize just something from one species and like just that thing? So if it's anti-rabbit, how is it binding to the rabbit protein? Like, what is it from the rabbit that's actually being recognized?
You know, I actually don't know, but I can find out and let you know via email, or we can talk a little bit after class and do a web search. What that would be like. Anyone else?
So the cell of interest that has to be rabbit, I mean from rabbit or from other species?
Another species, so not rabbit usually. You could use a rabbit antibody in a rabbit, but that's typically not used because then you'll have species interactions with the rest of the cells. All right, well thanks guys.
Any comments on the presentation?
There's a lot of jargon.
Sorry.
The Brett said that you were very calm.
Really?
Yeah, your tone was calm.
was like consistent.
I can't breathe. Okay. We good?
I think-
your answers to, sorry, I like your answer to Greg's question where you
Yeah.
tell them to look at them, because that's they always ask you things you have no idea. And like, they don't know how to use their book either. Have you found that? Yeah. So like, they won't look it up themselves and they'll never go, but if you let it go and help them look stuff up,
Yeah.
it's really helpful.
Yeah, that was definitely a good response.
Yeah.
One very minor suggestion is it's nice to use the board, but without a prior understanding, I'm not sure how much sense that would make
Right.
to anyone. So like, just asking, like anyone without a bio background, were you really able to follow what was on the board?
Probably
Not especially.
not. Yeah.
Right, like with a bio background it makes sense, but you know, just keep that in mind that you're teaching new concepts. Okay, thank you.